Hello everyone. My name is Yitao, and I come from the Tokyo University. And today I'm excited to introduce our work, and means STAIG, Spatial Transcriptomic Analysis via image-aided graph contrastive learning for domain exploration and alignment-free integrations. And here is today's my outline. First, I will introduce the backgrounds of the spatial transcriptomic data, and then is the overview of our method. And so finally, I will show you some of our experimental results for our tools. And as we know that spatial transcriptomic is a new technology, and it can capture the gene expressions, and at the same time, it can contain the high resolutional spatial context. And as shown in the pictures, it can collect, for example, for the Vision's platform, it is collected by the spot, and each spot can contain the spatial locations and the gene expressions. And then at the same time, it can contain the HE images. We know that for the now, it has already exists several spatial transcriptomic ST data analysis methods. However, they have some limitations. First is the bias in the static graph contractive structural contractive. We know that for the traditional method, we first to transfer their spatial locations into the graph, and they always build the graphs based on the some steps. First, for the we set each spot as a node, and they build the link, build each between the spots. And for one spot, it will have the several links with their surrounding spots. And once their graph structures is contrasted, they will not change during the trainings. And the next one is the limitations in the integration histology images. And this we will talk later. And the third one is that for the multi-slice situations, we need some extra alignment to first alignment their spatial coordinations and then for the integrations. And so consider this challenge, which we propose the stakes. It can extract the valid information from the images and without any pre-trainings. And it also can integrate the genes and the spatial data and the images informations. During the training, it can dynamically adjust the graph structures, and it can for accurate spatial domain identifications. And finally, we achieve the enable to end-to-end batch integrations and without any extra alignment before the using. And here is the overview of our stakes. And for the stake, it contains two main steps. First is the image information collection, and the next is neural network, deep neural network models. And the most distinctive pattern is this one. For the stakes, it first gets the spot images patches, and then we design a unique and the suitable processing steps for these pictures. And after that, we use self-supervised method to transfer the information into the embeddings. And based on their embeddings similarities, we transfer their information into the weights for each edge. And then we use contrastive learning method to get their embeddings. After we get their embeddings, we can achieve the batch integrations and the spatial domains identifications. Sorry, we're stuck. Okay, and here is one of our results for the integrations. As can show in these pictures, stakes can achieve several ways for integrations. And this is, for example, vertical silence and horizontal silence and partial overlap integrations. we can just to merge them together and input to the STAIG's models, and it can automatically to integrate them together. And the most important is that not only the same platform, the STAIG also achieves the cross-platform integrations. And for different platforms, we also can just to put them data together and achieve the better, greater integrated. We can see in the UMap, we can see even in the different platform, the same tissues from the different platform can get the well matched in the UMap. And next, I want to talk about the imaging features extractions for the STAIGs. And the STAIGs can extract imaging information in a self-supervised manner and without relying on the pre-training or the class labels, only relying on a simple input HE images. As we can see, this is HE images for our brains. And it's hard, it's hard to recognize, and it's also difficult for the machine to recognize which layers is different from each layers. And here is the manual annotations. We can see even we know the annotations, it's hard to split it by our human's eyes. So machines is also very difficult to find that. But under our STAIG's process, and we can see that STAIG can recognize some of the layers, and we are matched with the manual annotations. And for the cancels images, we also can say that for the cancels regions and the normal regions, we can see STAIG we are split. However, we compare with the existing the baseline methods like STLearn, DeepST, or ConST. They use maybe some of them use the RGB-based feature extraction or the CNN-based or the transformer-based. We can see that their features embedding is meaningless. They are not matched with the manual annotations. So their problem is that these features cannot really help to identify the spatial domains. And for the spatial clusterings, we use some matrix to apply the STAIGs to the common benchmark data sets, human dorsolateral prefrontal cortex, DLPFC. This is common benchmarks. And we can see that STAIG get the highest performance, and their cluster is well matched with manual annotations. And even without images, like other platform, the STAIGs also can have a good performance. We for example, like mouse hippocampus data sets in the slide sequence V2, we can see the STAIGs can recognize the major analytical regions, and particularly in the Emmons holes, and can separate them together in the CA1 and the CA3. Or for the star map, STAIGs can get the highest ARIs. And not that under the UMap, we can see the different layers will separate together. And with the baselines, we can see other methods, the UMap's different points are mixed them together. Yeah, sorry. And next is the case analysis for the STAIGs. We apply the STAIGs in the tumor micro environment. In the detail, we apply them to analyze zebrafish melanoma and to find a region called interface in the original studies. They define this special region that is in the boundary of the tumor and the muscles. And for STAIGs, we find that this kind of interface can further to divide into more small clusters. And in these two clusters, so one cluster is muscle proximal. They are more like their gene expression is more like a muscle, and for the other point, for the another cluster, they are more like a tumor. However, in this type of data for the other method, they need to combine with the single cell RNA data to find this kind of regions. However, use our STAIGs, we can directly use this just one data set to find this kind of 3 genes. And our work has been published in the bioRxiv, and the next is the QR code is our tools in the GitHub, and you can try our methods. And that's all. Thank you.
Does the audience have any questions for Yitao Yang? I'm pretty curious. You mentioned that you have tried to integrate data from different platforms.
Yeah, different platform.
Yeah, I'm actually very curious from the user point of view. So, how much does a different platform agree in terms of, let's say you have the same tissues, that if you use different platforms, how you have and how coherent is the data for different platforms? Have you done that kind of comparison before?
Yeah, we analyze that, and we think that this is based on the different technologies, and we know that for spatial transcriptomics, they have 2 kinds of technologies. The first is present with vision, and next like a slide or stereo or Murphy, they are different. And I think 2 kinds of technologies, their batch size is batch effect is very huge. But in the same technologies, even in the different platform, they can easily to integrate together, and their batch effects are not very huge, I think.
So you mean the same platform, the batch effect can be quite.
Yeah, it's more easier to integrate, even if they are based on the same principle of the capture the gene expressions. I think they will easy to integrate.
So any like with any other platform stronger one aspect, then like what are the predominant, like it's Murphy's and Bishop, are they complementary? That you think is complementary based on your comparison, or you think it's one is enough and it's very commercially commercial question?
Yeah, maybe I'm not very clear.
Yeah, I'm curious because it's always, even from my field itself, we always have even the same data. If you run the same sample again,
yeah,
you may not get a totally same kind of data set. And it's something that as a user, I will always be concerned with, how reproducible. Yeah,
okay. Now for the reproduce, I think the robotic for our tools is very good. Even in the for the same tissues, even from the different donors, it can the same. It also can get the good performance, I think. Or if this is one tissue and from different donors, can easily to integrate together, and we will imitate the batch effect, I think. Yeah.